Today, our competency is mechanism of urine concentration and dilution, countercurrent, multiplier and exchange. Actually, countercurrent multiplier and exchange, they together, both of these together form a system. We call it countercurrent multiplier system. Without this system, it is not possible to concentrate urine or dilute urine. Sometimes we concentrate urine, particularly at the time of dehydration. When you are not drinking water, so in the body there will be shortage of water, so this will lead to dehydration. Under this circumstance, to preserve the water content, kidney will produce concentrated urine. How? This is the mechanism. In instances when you drink more and more water, means overhydration. In that case, blood and tissue fluid becomes hyperosmotic. So, in such circumstance, to bring down the osmotic pressure to the normal, body eliminates through kidney excess water and involving this mechanism. So, this mechanism, countercurrent multiplier and exchanger system, they are actually responsible for concentrating the urine and diluting the urine. So we will study how these two systems, two processes are involved and where it is done. It is done concentration of urine or dilution of urine, or the place of countercurrent multiplier system is in medulla, not in renal cortex. And medulla where by the loop of Henle's of juxtaglomerular nephrons. There is no role of cortical nephrons, cortical loop of Henle's. The loop of Henle's of cortical neurons, they don't be involved in this process. Only the juxtaglomerular nephrons, they are having loop of Henle. These loop of Henle are very long. They penetrate up to the inner part of the medulla, that is up to papilla, the corner, the inner end of the pyramid. Okay. So these are the questions, possible questions from today's discussion. What are these? Countercurrent mechanism. And unlikely this sort of question to come, because the answer will be large, lengthy. Why? This countercurrent mechanism actually consists of other processes. What are these? Countercurrent multiplier system, countercurrent exchanger system, and the role of urea. So you have to describe these three things when only countercurrent mechanism is asked for. Okay? So you have to explain this thing through this and the role of urea. Okay? So this process would involve these sub-processes. Okay? But these are important, very important. For 5 months, this can be asked. Countercurrent multiplier, countercurrent exchanger, and this can be is the most frequent question. What is this? The mechanism of concentration of urine or mechanism of formation of diluted urine. Okay? And formation, mechanism of formation of concentrated urine, role of urea in formation of concentrated urine. So these are the, and for 3 months this can be asked. Urea, diuresis, the difference between osmotic diuresis and water diuresis and loop diuretics. So these are the usual questions asked for from this today's competency. Okay, let us proceed. So to understand the countercurrent mechanism, countercurrent system, we must know something. One point I said, loop of Henle. Who is loop of Henle? Juxtamedullary. They are having long loop and they extend up to end of the medullary pyramid to the papilla. Okay? So they extend up to that. And whenever necessary, this system concentrate the urine means it makes the urine hypertonic in relation to blood or body tissue fluid. So it is hypertonic. So it is, and the hypertonicity of the urine may go up to 1200 millimole per liter. Maximum, this much we can concentrate. When is this situation arises? When we are dehydrated. Okay, so in such condition, urine concentration or osmolarity may increase. Normally it is how much? It is around 600 milliosmol. Osmotic pressure of urine. So it normal. But in such situation, dehydrated condition, it may go up to 1200. But whenever you are overhydrated, then what will happen? The urine will be diluted. Means in this urine, the solute concentration will be less. And definitely osmotic pressure will be coming down. How much? It may come down to near about 60 milliosmoles per liter. Okay. During dehydration, as I said this point, and another thing, the components of the concentrating and diluting system. What are these? One is the countercurrent system. As I said, another is countercurrent exchanger and urea cycle, not the metabolic part. The cycle means from collecting duct to interstitium to this vasa recta. You don't know what is vasa recta? Huh? Who knows vasa recta? What is vasa recta? Juxtaclomular nephrons. These nephrons. So they are also having ascending and afferent and efferent arteriole. This efferent arteriole, after coming out of the nephron, they produce what? Capillaries. Now these capillaries run along what? Loop of Henle and in reverse direction. Okay, suppose loop of Henle is sending filtrate towards the cortex, but blood will be passing in this counter way. Okay, and loop of Henle is coming down through means this descending limb of loop of Henle. Here, what will be there? The vasa rectus will be going upward. So all are in their counter position. The vasa recta, they are in counter position. Similarly, the circulation is counter way, and similarly, in the loop of Henle also coming down and going up. Okay, so there will be a unique process by which kidney makes the urine concentrated. We will discuss the mechanism. So this countercurrent system, it is, as I said, it is having mainly two parts. One is countercurrent multiplier, means it will multiply, it will gradually increase the osmolarity of the medullary interstitium. What is medullary interstitium? In the pyramid, in the medullary pyramid, you have tubules. These tubules are what tubules? Vasa recta tubules and also loop of Henle. Without this, we have there collecting tubule. Hello. Without there, we are having collecting tubule. So in between, there are some other tissues. These are interstitium. These are medullary interstitium. So in the medulla or medullary pyramid, what you will get? You will get collecting duct. You will get loop of Henle, both descending as well as ascending. You will also get vasa recta. Beside, there are interstitial cells. There is interstitium. Okay. Through figure, I will demonstrate this. And this system, countercurrent system, particularly the system, the total system will include this thing. What are these? Descending limb of the loop of Henle of which nephron? Juxtaglomerular nephron. Okay, not cortical. Okay. Cortical nephron don't produce vasa recta. Okay, they don't produce vasa recta. So, descending limb of the loop of Henle and thin and thick segment of ascending loop. The ascending loop of Henle, initial part is thin and the upper part is thick. Okay, so ascending limb has two parts. One is thin part, another is thick part. Thin part is passively soluble to sodium ion. Sodium chloride can passively pass from the luminal filtrate into the interstitium. But when it goes up, enters into wider part, thick part, then it is not passively transported. It is now transported, sodium chloride is actively transported. We will discuss. Okay. Then, what is this? The medullary interstitium, distal convoluted tubule, collecting duct and vasa recta. Vasa recta is having ascending and descending part. So these are involved in the system. Okay? Without this system, not possible to concentrate the urine or to dilate the urine. Now see, this is the cortical neuron, nephron, and see it is loop of Henle, very short. And this is the juxtamedullary nephron and is and is descending. So this is descending and descending, this is loop of Henle, and now ascending loop of Henle, yellow one. This is thin part, this is the thin part, thin descending and thin ascending, then thick ascending, and it is passing through this. Sorry, I am wrong. This way it comes. This way, this way, this way. This is this way, and this. So, collecting duct. So, in between are there medullary interstitium. These are the medullary, the gap are made up of medullary interstitium. Okay? And this is the junction. Cortex, medulla, juxtamedullary junction. Okay. Again, a little bit explained. So here you can see this is the efferent arteriole, efferent arteriole. So it will enter into the medulla and form this. What is this? This is vasa recta. They will be aligned here along this tubule. They are fitted here. Okay, and this is the collecting tubule, papilla, urine is formed there, entering into minor calyx, then into major calyx. Okay, now you can see one thing here. In the medulla, the osmolarity. See, the gradual increase in osmolarity in the interstitium. Interstitial is not shown in another figure I will show. Here also, in the if it is 980, in the interstitium will also be 980. Here it is 1200, in the interstitium will also be 1200. If here it is 580, here is also 580. Similarly, here it is 300, 300 this way. We can see the osmolarity from the cortical area, juxtamedullary area, as you go downward, gradually the osmolarity is increasing. How this is increasing? What is this process? This is done by countercurrent multiplier system. It is multiplying the concentration of the solute in the interstitium. First, it is changing the osmolarity within this. As a result, what will happen? This will be coming in equilibrium, isosmotic. Here, if it is here 760, here also the interstitium should also be 760. Okay. So, this is the countercurrent. I will explain. See what is happening. See what is happening. What is this? Whether it is descending or ascending? Tell me. Whether this tube, loop of Henle, whether it is descending or it is ascending. Descending from here, proximal tubule, now it is coming into the medulla, and you see, initial part is thick, then it is thin. And it forms the hairpin. It forms the hairpin, and you see, this is the what I say in between, what is the interstitial tissue. Now, what is the osmolarity of the interstitial tissue? Starting from 300, gradually increasing. And at the tip of the papilla, up to the tip of the papilla, when it reaches up to, it becomes 1200 milliosmol per liter. So how this gradually increasing osmolarity is taking place. And it is most important for developing concentrated urine. And also you see, this is the glomerulus, afferent arteriole entered into the glomerulus, efferent arteriole coming down, okay, and then coming up. See, and now joining the bed. See, whenever this is changed, here also same change is happening within these blood vessels. Same change is taking place. Why? Always we know osmosis. So isosmosis. When there is hypertonicity, so it will try to make the hypotonic, isosmotic. So that's why if one part is hypertonic, then it will be trying to be isosmotic. Okay. So now you can see the color of this color is gradually concentrating. Here it is gradually fading. It means the osmolarity is gradually increasing. So the countercurrent multiplier system has only one function. What is this? To make this gradual increase in osmolarity. This is its function. It is the function of the countercurrent multiplier system. Okay. See, if you leave this area, medulla, concentrate on cortex, you will find the osmolarity everywhere is same, 300 milliosmol. Everywhere it is same, either in the blood vessel or in the filtrate or in the tissues of the renal cortex. Everywhere it is 300. But as soon as it enters into this, gradually its osmolarity is increasing. Here it is maximum. Again, when the fluid goes out, the osmolarity gradually decreases, and not only this, it becomes hyposmotic. 100 is hyposmotic. So there is one other important point you will be here to mention. So when the fluid is entering, that time it was initially 300. But when it gradually advances, its osmolarity gradually increased up to, it may go up to 1200. Again, when it goes up, it will be coming down, and when leaving the loop of Henle, okay, and passing through this is thick limb of loop of Henle, and from here starts the distal convoluted tubule. This distal convoluted tubule has two parts. One is the early part and is the distal part. Both this part, this diagram explains countercurrent exchanger system, not the multiplier system. Though it is there, the multiplier is also there. So how these both multiplier and exchanger are coupled. So this coupling is shown here. How this is made? Gradual hypertonicity, gradual osmolarity from 300 to 600. Let us concentrate on this. I will explain the mechanism. Okay? For the first instance, there is no countercurrent multiplier system. Okay, what will happen? Filtrate will enter into the loop of Henle. Okay, we'll fill it up. We'll pass through this and fill it up. And what is their osmolarity? 300. Okay, now this tubule and this tubule, their function is different. One opposes the other's function. How? This is permeable to water. This descending limb of Henle is permeable to water but impermeable to solute sodium chloride. Okay? But this lean, ascending lean, is impermeable to water. This is permeable to water, this is impermeable to water, but permeable to water but permeable to sodium chloride. Okay? So this is the situation. Now everywhere osmolarity is how much? 300. Okay. Now what happens? If I keep this like this, what will happen? From here, sodium chloride will be diffused here. From here, sodium chloride, because of they are permeable, this limb is permeable, so it will be diffusing. Suppose 100 unit is diffused. So now what will happen? It was initially 300, 100 passed out, so this will be 200. Initially it was 300, out of this 100 milliosmol transferred to the interstitium. So interstitial, it was previously 300, now it will be getting 100, so it will be now 400. So here it has come to 400 and it has reduced to 200, and there is a gap of difference of 200 milliosmol, 200, 400, 200, 400. So means this is diluted, this part is diluted and this part is concentrated. Why it happened? Sodium chloride is absorbed. This part it is absorbed passively and from this part it is actively. Ultimately what is there? This became, when this it is leaving, see it entered with 300 milliosmol and going out with 200 milliosmol diluted. We said ki when it will be entering and with some osmolarity, and when it will come it will be less. Means it will be hyposmolar. So it is now hyposmolar. Okay. Now I'll do one thing. What? After some time, this area became 400, means it is hyperosmolar, more concentrated, whereas the fluid is less. So what will happen? Fluid, this is permeable to water. So water will come to make it equilibrium, make it isosmolar. So from here, water will be entering. Okay? Water will be, so this will become concentrated gradually. This will become concentrated and will become 400. See, it has become iso. Okay? Now, it is very easy to understand, otherwise it is very complicated. Very easy to, for better understanding, follow the only book, that is Guyton. Go through 2 times, 3 times, it will be clear to you. Okay? For everybody, you just follow these books. So now we see, because of active transport or passive transport of sodium chloride here, its osmolarity increased to 400. Okay, but it was 300, so they have to be similar, and it will make this area also 400. Now they are isosmolar. Okay. Now this is stable. Now what I will do? Another bout of fluid I am sending into the tube. Another bout, some more glomerular filtrate I am sending. Let's see what is happening. We'll see osmolarity is further increasing. How? Here I send 2 units, 2 units of 2 segments of filtrate. This is that 2 segments of filtrate. And because this area is occupied by these 3 segments, they will be going forward. This area will be pushed to this area. So all this 400 will be coming here. How many 400? 1, 2, 3. 1, 2, 3. Okay. So 1, 2, 3 coming. And as a result, what is happening? See what is happening now? Again, from here, the sodium chloride will be coming. From here, because these are passively transferred, this will be actively transferred. As a result, this situation will be there. So it is now step 4. Yeah. So more sodium ion is entering. As a result, it is becoming hyperosmolar. And after some time, this area will also be 500, 500, 500, 350. Because it is diluted by this. And why this became, from here, why it is increasing? Why it is increasing? This side is increasing. Because this side is permeable to water. So water will come out. As a result, this will be concentrated. Isn't it? So its osmolarity is increasing. And if the process is repeated for 4 to 6 times, ultimately osmolarity will be multiplied into this. Here it will be 1200, 1200, 1200 like this. So in this way, the osmolarity is gradually increased from here towards the apex, 300, 700, 1000. So this is the way, mechanism by which increased hyperosmolarity, increased osmolarity means hyperosmolarity developed by the countercurrent multiplier system. What is happening in different segments, that has been explained here. Now see, when it was it has entered the final condition, it is 400, it is 1200. Now it is ascending, gradually decrease, the osmolarity will be decreasing, and when it will be leaving, it is becoming hyposmolar. So this hyposmolar filtrate will be passing through the descending, distal convoluted tubule. So in the distal convoluted tubule, this is actually, this is what? Collecting. This is the collecting duct. This is the distal convoluted tubule. Okay. This area passes through JG apparatus, through this junction. After coming out of that area, there is going towards the collecting tubule has 2 parts. One part remains in the cortex, and the bigger part, most part remains in the medullary region. So here, what happens? The diluted filtrate is coming, more hypo. Now it is entering here. It is entering, coming to join the collecting tubule. Here I said the permeability to sodium ion is variable. Also water variable. These are modified by 2 things. One is aldosterone, another is by ADH. This part, the initial part, has no role for aldosterone. The early part of the distal convoluted has no role for aldosterone. But later part has the role. We know what is the function of the aldosterone. They increase the uptake of sodium ion, and since sodium ion is coming, water is freely coming with sodium ion. So there will be, see absorption of water, also sodium, potassium and chloride, because there are sodium, potassium, chloride exchanger. Sorry, co-transporters. Okay, so with this co-transporter, sodium ion it will be entering. But collecting duct. This is the late part of the distal cord DCT, later part, and this is the early part, the initial part of DCT. This part, the epithelial cells of these epithelial cells are not responsive to ADS. They are not responsive. They don't have any receptor for aldosterone. But this part is having the role. These cells and these cells are responsive to aldosterone. Aldosterone here can act on it. As a result, what will happen? There will be sodium chloride coming out into the interstitium, and water will also be coming out. Here, since it is permeable, here what happens? This area is strictly impermeable to water. Only who can make it permeable? ADS. Only ADS, by placing aquaporin, through which water comes out of the filtrate. So they can also, these cells are under the influence of ADS. Whenever there is lot of ADS, so when any water will be passing through, they will be absorbed into the interstitium. All water will be absorbed into the interstitium. Okay? Under the influence of ADS. And here what happens? This is done under the help of aldosterone, also ADS. Okay? So now this tubule entering into the medulla. So this part now entered into the medulla. Here what will happen? Water will only come out of the filtrate if there is ADS, otherwise not. Okay, so water will come out under the influence of Acha. Now what thing was, if lot of water comes out of the filtrate in the collecting tubule, from the collecting tubule, what will happen to the urea concentration? Here some urea is there. So I am taking out the water. So urea concentration will be rising or falling? Rising. But here its concentration is less. So there is difference. So there will be coming out. And this has got one advantage for the interstitium, because this urea will increase the hypertonicity. So they further help in increasing the hypertonicity at the medullary region. Okay, so these are the role of urea. Okay, now let's say this is the exchanger. What is the situation in the interstitium? Lot of sodium. It came out from different limbs. From a lot of sodium, hypertonicity is made because of sodium. So, lot of sodium is there. This sodium, now blood flow. This is the vasa recta. Vasa recta is entering into the interstitium. Okay. So its osmolarity was 300. Okay, so when it will be coming down through this. Since this outer area is hypertonic, so water will come out. Water will come out. When it is entering, okay, water will come out and sodium chloride will be entering. As a result, here also osmolarity will be 1200. But when it is coming, will be flowing throughout these, here also what happens? Sodium chloride, now it will be gradually hypotonic. It has to be, because this area and this area must be almost same. When it is passing throughout this, it is becoming concentrated. How? Entry of sodium ion and exit of water. So it is happening there. Water is exiting, sodium chloride is entering. Okay, urea is altering. As a result, its concentration is increasing, osmolarity increasing. Here it is becoming maximum. Now, while it is going up, the vein, vasa recta, it is going up. Then what happens? From here, sodium chloride is coming here, and water is entering. So this is the same thing. See, here water is entering, here it is coming back. Ultimately, water content and sodium chloride content in the interstitium remains unchanged. And this is the function of this. So, what is the function of the exchanger? To maintain the hyperosmolarity of the medullary interstitium. This is the only function. So, keep it in your mind. What is the function of the exchanger? To increase the osmolarity of the interstitium of the fluid as it goes down. Okay, from cortical region to inner medulla. Okay, so its function is to make the interstitium gradually increasing hypertonicity. And what is the function of this? To maintain this hypertonicity. To maintain this otherwise what will happen? By the initial formal process, exchanger system, they will gradually increase. Okay, there is a limit, so that the hyperosmolarity that is created by the multiplier, and this is maintained. After some time, it is maintained. We know it is maximum, we can increase the hyperosmolarity up to 1200. 1200 to 1300, not more than this. So, then the whole system will be, otherwise the whole system will be non-functional. Okay, so this is the function of the exchanger, and you can see the function of the role of urea also. Urea also helping, what it is helping? Helping to create the hyperosmolarity. Nearly 30 to 40% of the hyperosmolarity that has been created is contributed by urea. Understood? So this has to be remembered by you. If you study today itself, countercurrent, it will be to you, it will not be very complicated to you. I will suggest you, advise you, today itself you go through this book. This is how both the system operates. Very clear understanding. If you minutely observe this, you will be. I think this image is not available in your book, in any of your book. The same thing. Role of urea. This is. What is diuresis? What is diuresis? Diuresis, increased urination, increased water loss. Okay, this is called diuresis. ADS, what is the function of ADS? Uptake of reabsorb the water. So it is acting against diuresis, isn't it? That's why it is called antidiuretic hormone. It reduces It reduces secretion of, not secretion, elimination of water from our body. Okay. The system will try. What is the required amount? This is the requirement. Okay. I always try to maintain this. If it is more, I will increase the excess water in urine. And if it is becoming less, then through urine I will not pass water through this. So I will absorb it. So depending on the hydration condition of the water in the body, it will decide what to do. Either to concentrate the urine or to dilute the urine with the help of the hormonal system also. This system as well as the application of ADS and aldosterone. What is diuresis? Diuresis is an increase in the rate of urine output. Whenever this term comes, so diuresis in the hypertensive patient, or who are having edema, peripheral edema, what type of edema, or there is ascites. So this drug is given. ADA means the drug, a diuretic drug is given. They will block the function of the ADS. Okay? They will act on the receptors. ADS receptor. As a result, what will be? Lost. The excess fluid will be lost. Okay? So this type of drugs, they are called antidiuretics. Okay? Antidiuretics, because they prevent diuresis. Okay? Now, there are two types of diuresis. One is water diuresis, another is osmotic diuresis, and there is also pressure diuresis. Okay, what is water diuresis? Increase water intake, decrease osmotic pressure everywhere, pressure will be decreased. As a result, there will be decrease or increase ADS. Why not increase? If only in my body already water is high, if I secrete ADS, it will further absorb water from the urine. As a result, more water content will be more. That I don't want. That's why I will inhibit the release of ADS. I will inhibit in the, where from ADS is released? Where from? What is the source of ADS? Anybody? Hypothalamus. Hypothalamus, they are having osmoreceptors. What? When the osmolarity of the blood or tissue fluid is decreased, means more water. So these osmoreceptors get stimulated. There is hypoosmolarity, means there is more water in my body. So they stimulate, actually they inhibit the hypothalamus, don't secrete ADS. It inhibits secretion of, or don't, I will not say secrete, don't synthesize ADS. ADS is secreted in posterior pituitary. Synthesized in their function is to posterior pituitary only release it. Stimulation has to come to hypothalamus for release. And how the stimulation will be coming to the posterior pituitary? Through a tract. Through a neural tract. Do you know the tract? Hypothalamico-hypophyseal tract. This is a neural tract. Since this is a neural tract, its cell body is in hypothalamus, supraoptic paraventricular nucleus. So their nucleus is there. This nucleus, cell body, they are preparing this hormone, ADS. Now when it is stimulated, now this ADS through exoplasm will go to the posterior pituitary and they will be released from there into the blood. Not directly into the blood, to the ECF. From the ECF, it will be absorbed into blood. Now it will be circulated and will be coming to the kidney. Okay, so remember this. Hypothalamic, you must have known this. Hypothalamic, not only this hormone, another hormone. All the posterior pituitary. What are the other posterior pituitary hormone? Vasopressin, LDA, same name. Previously, we used to call vasopressin. Previously, we called vasopressin. The person who identified this, he first observed that it is having some vasoconstricting effect. That's why name is given as vasopressin. Thereafter, it is found no. Its main action is on kidney to absorb more water. Okay, more water absorption, actually blood pressure will increase, no? More volume will be there, more intake, blood is increasing because of entry of, for temporary period, blood pressure is increasing. And it will not be, within 40 minutes, again it will be coming down by the action of this. Okay, so, what is the other hormone? Oxytocin. Anybody know the function of the oxytocin? Parturition. Okay, so you know that. So this will be inhibited. Why? Because of decreased osmotic pressure. Why it has been decreased? Because of this. Okay, now decrease in. There are two types of water reabsorption. One is facultative, another is obligatory. Facultative means in some portion of the renal tubule where only in collecting duct and in last part of the distal convoluted tubule. That part, if it wishes, it can absorb. When wish develops, when ADS is there. So that's why this part is called facultative. Water is reabsorption. From this part is called facultative water. And other part is, what I said? Obligatory. Obligation. What obligation? Because water is permeable. Wherever it is permeable, and here osmotic pressure is low and here is high. So water has to pass if it is permeable. This is an obligation. This is the obligation. So this is the obligatory water. Two types of obligatory and facultative. Okay. Then water reabsorption, urine, large volume of blood. So this is the water diuresis. And osmotic diuresis, the difference. Reabsorbable solute in proximal convoluted tubule cannot be absorbed. As a result, decrease in obligatory H2O as reabsorption, decrease in sodium concentration in the renal tubule, and decrease osmolarity medullary interstitium, decrease facultative water reabsorption, urine, large volume of isotope. So this is the difference between the two. Okay, thank you.